In this video, we're going to generally be talking about the way genes are regulated in both prokaryotes and eukaryotes with a focus on prokaryotes. So some proteins are constitutive, which means that they're always needed for survival of the organism. And then we also have genes simultaneously which are inducible. So when the cell doesn't necessarily need them, it won't be wasting energy to express those genes. Those genes would be inducible proteins or inducible genes, where they're only expressed when needed. So we're not wasting energy creating things that we're not necessarily using. So they're only made when needed. So constitutive genes are always going to be needed, therefore they're always going to be expressed, whereas inducible genes or proteins are only going to be made when needed. So some ways that eukaryotes could regulate their gene expression is one, they could inhibit their gene expression, inhibit DNA expression. So inhibit RNA polymerase from coming and creating a transcript. Now we could also break down mRNA, messenger RNA, or the protein itself. So we can hydrolyze, or we know that hydrolysis is the breakdown of something. So we could break down that messenger RNA, or we could break down that protein. We could also inhibit transport of that mRNA, that transcript, out of the nucleus. So if we don't modify that messenger RNA properly, it won't ever get sent out of the nucleus, and if it never gets sent out of the nucleus, it can never get picked up by a ribosome to get translated. And then we could also regulate our translation control, so we could do things like inhibit our ribosomes. So a lot of different ways that eukaryotes will regulate gene expression, gene expression. Okay, so we have two terms here. We have positive regulation, or what we would also call upregulation, and that's the process by which a cell will increase the quantity of a cellular component, so like the RNA or the expression of the protein, in response to some kind of external stimulus. Now, the opposite of that is going to be negative regulation or downregulation, and that's going to be the process that's opposite of that, so which a cell will decrease the quantity of a cellular component, such as RNA or protein, in response to an external stimulus. So let's just say, an example of like downregulation would be, you could say that it normally has receptors for some kind of ligand or thing that would normally bind to the receptors, and let's say that there's this oversaturation. There's too much of that substance that's constantly binding to the receptor site on a cell. Well, what might happen is that cell will prune away receptor sites so that it's stimulated less to kind of maintain, you could say, homeostasis for the organism. So too much of a good thing isn't always good, and that would be, an example of a cell kind of reacting to some kind of external signal. Same thing with positive regulation or upregulation, let's say. And these are very niche examples. This isn't the only things that could cause upregulation or downregulation, but let's just say, there's an absence of stimulus, an absence of ligand being produced or existing outside of the cell, which is scarcely binding to the receptor site on the cell. Well, maybe what the cell might do to increase its receptiveness to that ligand is it might express more of the receptor site, so there's a higher chance of that ligand binding to the receptor. So that would be an example of like upregulation in response to the environment. All right, so now let's focus a little bit on prokaryotes. So prokaryotes, a commonly used example for gene expression in prokaryotes is called the lac operon. So all an operon is a cluster of genes. So we'll say Z, Y, and A gene. So Z, Y, and A gene, plus some kind of sequences nearby. So we're going to have I, P, lac, and O, which we'll talk about. So the lac operon is a very commonly studied operon in prokaryotes, and it consists of 3 lactose metabolizing enzymes in E. coli. So just to break down some of these colors here, you have a promoter for a regulatory gene. What's a regulatory gene? So it's going to be some kind of gene that's going to modify the expression of the main genes, the genes that we want to express when we're in the presence of lactose, for example, something to break down lactose. So the regulatory gene, so I being the regulatory gene, is going to code for a repressor protein. So as we could probably infer from the name, a repressor protein is probably going to repress something, prevent something from being expressed in this case. And then you also have the promoter. So kind of like we talked about before, promoters are going to be stretches of DNA that are attractive to your RNA polymerase. Your RNA polymerase will typically bind to the promoter to begin transcription. So you've got one, the promoter for the regulatory gene. So this is the repressor protein. And then we have another promoter for the structural genes. So the genes that we would express in response to having lactose build up inside of the cell. So E. coli could potentially break down lactose when it's within the cell, and it does so by lactose actually stimulating the expression of genes that would break it down or metabolize it. But the thing is, if there's no lactose present, you don't want to be wasting energy expressing the genes to break down lactose. So that's why we would call this, this operon kind of an induced operon or induced set of genes. So we have the promoter for the structural genes. So these are the main genes that we're looking for to break down lactose. But between that promoter and the genes of interest, we have this thing called an operator. So an operator, which we'll probably talk about in another slide, an operator is something that could potentially prevent, or it's a site where you're going to get something to bind to it, which could potentially prevent RNA polymerase progressing through with transcription to express these genes. And then Z, Y, and A, we don't need to really know the specifics, but we have beta-galactosidase and then beta-galactoside permease, which is going to actually be, it's going to create a channel so that lactose can enter. And then beta-galactoside transacetylase. These are all genes that would be associated with breaking down lactose and bringing lactose into the cell. So these are the genes of interest. Operator is going to prevent, the operator is going to be a binding site for a protein that's going to prevent transcription. Promoter is going to be where RNA polymerase would normally bind to in order to progress with transcription. And then you have these regulatory genes nearby. But we'll get into the details in a sec. So the promoter, that's going to be the site for RNA polymerase binding. Again, your operator is going to be a site for repressor binding. So repressor, remember repressors are probably going to repress something, probably going to repress the progression of RNA polymerase. So it blocks RNA polymerase. And so your repressor binds to the operator. And then you have this thing called a cap site. So that's going to be a site for cap binding. What is cap? Cap is a catabolic activator protein. So what it does is it allows RNA polymerase to bind to the promoter. So one thing about prokaryotic cells is that RNA polymerase needs more than just the promoter to bind to DNA. It also needs the cap bound to the cap site. Cap bound to the cap site. So here's the protein, cap, and then here's the site that it would bind to. And so in order for RNA polymerase to progress with transcription, cap needs to be bound to the cap site. So how do we get that to happen? Well, before we talk about the cap site and cap and blah, 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 let's talk about some more intuitive stuff. So what makes this intuitive is we only want to express genes associated with the breakdown of lactose if lactose is present. So that's pretty much all this is saying. If there's no lactose, when lactose is absent, the repressor binds tightly to the operator. So repressor is going to bind tightly to the operator so that when RNA polymerase eventually binds to the promoter, well, it can't progress. This is a way of inhibiting those genes from being expressed if there's no lactose. The repressor binds to it. However, in the presence of lactose, what happens is when lactose enters the cell, it's eventually converted into allolactose, which is just a rearranged isomer, and that's going to bind to that repressor. So here you have the repressor with this little opening spot, a receptor for allolactose. So when lactose enters the cell, what happens is the repressor is now dislodged from the operator so that RNA polymerase can progress with transcription. So I'm just going to break this down really quick here. So we have no lactose, which leads to repressor binding, which leads to the inhibition of RNA polymerase. We'll just say RNA, and I'll just put Pol for RNA polymerase. We'll just say inhibited. And what I mean by inhibited from progressing through with transcription. Now, or I guess in this case, it is inhibited in this case. It's denoted by this X here. All right, so now if there is lactose, so we'll just put a little arrow. So high lactose. That will lead to the repressor unbinding from the operator. So the repressor freed from operator, which will eventually lead to RNA polymerase progressing through with transcription. RNA polymerase, and we'll say RNA polymerase we'll say transcribes genes associated, let's put a soak, associated with lactose metabolism. Okay. So let's move on to the next slide, because there's not only one thing that needs to be present for this to happen, for transcription to happen. So cyclic AMP. Cyclic AMP is a signaling molecule that is expressed usually when we have low glucose. So it says here is expressed in an inversely proportional way to glucose. So if we have high glucose, we'll have low cyclic AMP. If we have low glucose, we will have high cyclic AMP. It's a signaling molecule. So in this case, actually, we'll start off here. So low glucose, it starts off with low glucose. So low glucose, and I'll just put GLUC. Hopefully you get the point. So low glucose leads to high cyclic AMP. So high cyclic AMP. Cyclic AMP binds to the cap protein, which binds to the cap site. So high cyclic AMP binds to the cap protein, binds to the cap site, and that allows RNA polymerase to bind to the promoter. So low glucose, which leads to high cyclic AMP, which leads to, you could just say active we'll just say cap for cap protein, which binds to the cap site, binds cap site, and then we know that when RNA polymerase binds to the promoter, it can progress with transcription. So RNA pol binds, we'll say binds promoter. We're running out of space here. All right. So low glucose leads to high cyclic AMP. High cyclic AMP leads to the activation of the cap protein. That cap protein binds to the cap site, and when it binds to the cap site, you're able to recruit RNA polymerase to bind to the promoter. So now let's talk about high glucose. What happens if we have high glucose? High glucose, high glucose leads to low cyclic AMP. CAMP. So low cyclic AMP. When we have low cyclic AMP, your cap protein is not active. So that leads to we'll just say non-active cap for cap protein. So your cap site is vacant, and if your cap site is vacant, no RNA polymerase binding to promoter. No RNA pol promoter. Let's put promote binding. All right. So hopefully that makes sense. Now, both of these things have to be satisfied in order for the expression of the appropriate genes. So let's just go back a little bit. So not only do you have to have lactose present, in order to express these genes, you need both of these criteria met. You need lactose present. That's a weird artifact. And you also need low glucose present. So high lactose, low glucose leads to the lac operon being transcribed and translated, being activated, you could say. So both of those criteria need to be met. So just to kind of compare, eukaryotes versus prokaryotes before we get into the eukaryote regulation stuff. So eukaryotes typically have a double helix in multiple pieces, meaning multiple chromosomes. So double helix DNA with multiple pieces or multiple chromosomes, and comparatively larger in our genome size, so more genetic material. And because we have a nuclei, we've got our transcription and our translation physically separate from one another. Our transcription is going to happen within the nucleus, and then those mRNA transcripts are going to be sent outside of the nucleus for the translation portion. And then each gene is going to typically have its own promoter, for the most part, in eukaryotes. And the genes are going to be far apart. There's going to be a lot of regulatory components of our genome in between our genes that we're actually expressing. And then 3 types of RNA polymerase whose binding depends on other proteins. Now, when we're talking about prokaryotes, we know that generally prokaryotes are going to have circular DNA. They're going to have a single chromosome, and they might have other plasmids, which are just smaller pieces of circular DNA. Genome-wise, genome size is going to be comparatively smaller, and transcription and translation are not going to be separated. Remember, prokaryotes do not have a nucleus, and therefore, as it is transcribing a gene, for example, ribosomes are already going to be picking up that transcript and creating the protein. It's going to be translating that mRNA as it's pretty much being pumped out of RNA polymerase. And then typically with our prokaryotes, it's going to be more common for one promoter to activate several genes that are usually related to one another. So your genes are going to be clustered together, usually by function. And then one RNA polymerase that does not depend on other proteins for binding. So this is going to be our RNA polymerase. One type of RNA polymerase.